Hello friends and welcome to CEC live lectures. In the ongoing lecture series on plant hormones, we have already discussed about some of the important regulators like auxins, cytokinins, gibberellins, abscisic acid, ethylene and brassinosteroids. So in continuation with the lecture series, today I am going to start with yet another important plant hormone, that is jasmonates. Now in this lecture we will discuss that what are jasmonates, what is their chemical nature. Then I will give you a brief overview about the history of jasmonates, that how these jasmonates were discovered and during which time they were declared as plant hormone. Then we will discuss about the biosynthetic pathway of jasmonates, that how these important regulators or growth regulators are synthesized in the plant system, followed by their signaling mechanism, how they exhibit the effects in the plant system, and in the end we will discuss that what are those effects that are exhibited by these important bioactive molecules. So to begin with, what are jasmonates? These are nothing but the oxidative derivatives of unsaturated fatty acids, or PUFA or polyunsaturated fatty acids, and these fatty acids undergo enzymatic oxidation to produce oxylipins, and jasmonates are nothing but an example of oxylipins. Now these fatty acids, they are acted upon by oxidative enzymes like lipooxygenase, and they get converted into oxylipins. Apart from this enzymatic degradation, these fatty acids through non-enzymatic pathway sometimes also get converted into oxylipins or jasmonates. Now jasmonate is a collective term which is given to different derivatives of jasmonic acid, and these derivatives can be methyl jasmonate or jasmonic acid in conjugation with different type of amino acids or isomer. Now in 1962, for the first time, De Molay and his group, they isolated the methyl ester of jasmonic acid when they were working on a flower known as Jasminum grandiflorum. And this was the first instance when the scientist reported the presence of jasmonic acid derivative in the plants. And subsequently in 1980, for the first time, the effect of jasmonic acid onto the plant system was reported, and in that scientists report that jasmonic acid has a effect on the senescence of the flowers, that is, it promotes the senescence of the flowers and thereby inhibit the growth of the plant. So this was the first report when the scientific community reported that it has an inhibitory effect on the plant growth or the plant system. And subsequently in late 1980s or 1990s, the first indication of the altered gene expression was reported, wherein it was mentioned that jasmonic acid or its derivative not only have a physiological effect, but they cause alteration in the expression of the genes that lead to those effects. Then in 1984, the biosynthetic pathway of jasmonic acid was elucidated by Wick and Zimmerman, wherein they stated that the biosynthesis of jasmonic acid or its derivative takes place either in chloroplast or also in the peroxisome. And they further reported that alpha-linolenic acid is the major precursor for the biosynthesis of the jasmonates. So it all started with alpha-linolenic acid, and as you can see that PUFA in the plant system or polyunsaturated fatty acids in the plant system, they are acted upon by LOX enzyme, that is lipooxygenase enzyme, thereby producing various downstream molecules. And amongst these molecules, jasmonates are the molecules which we are mainly concerned with. So linolenic acid is released from the membranes or the plant cell membranes or the chloroplast membranes, because we know that during stress conditions there is a lot of generation of these free radicals and activation of certain hydrolytic enzymes. And these enzymes act on the membranes of the cell, thereby degrading or disturbing their integrity. And as soon as this integrity is disturbed, certain fatty acids are released from the membrane, which are acted upon by the hydrolytic enzymes which are activated during these oxidative stress conditions. And one such enzyme is 13-lipooxygenase enzyme, which acts upon alpha-linolenic acid to produce 13-hydroperoxy octadeca trianoic acid, that is 13-H-pot, which is an important intermediate, first intermediate that is formed during the biosynthesis of jasmonates. Now this 13H pod undergo dehydration with the help of an enzyme known as allene oxide synthase, and it produces an unstable intermediate which is known as 12,13 oxido 9,11,15 octa decatrienoic acid, that is 12,13 EPOD. And this unstable intermediate immediately gets converted into a cyclic compound with the help of allene oxide cyclase, and this compound is known as 12-oxo phytodienoic acid or OPDA. Now OPDA is another important intermediate during the biosynthesis of linolenic acid, and up till OPDA all these steps takes place in the chloroplast region of the cell. Once OPDA is synthesized, it leaves the chloroplast and enter into peroxisome region, where it is further acted upon by 12-oxo phytodienoic acid reductase enzyme, which causes the reduction of OPDA and leads to the production of 7-isojasmonoic acid. And this 7-isojasmonoic acid is produced from OPDA through a series of beta oxidation steps, wherein with every step 2 carbon molecule is detached from the parent OPDA, which eventually leads to the production of jasmonic acid. Now as I told you that jasmonic acid is present in different derivative form in the plants, and collectively all those derivatives are known as jasmonates. So once this 7-isojasmonic acid is produced, and because it is a very unstable molecule, and it immediately epimerizes to a more stable trans form of the jasmonic acid. So in the end we get a trans jasmonic acid, which further undergo different modification reaction. Dehydration is there, decarboxylation is there, oxidation is there, hydroxylation is there. So through these modification reaction, this jasmonic acid produces different jasmonic acid derivatives that are present in the plants. And these are the various modification reactions, like conjugation, wherein jasmonic acid molecule attaches itself with a amino acid, and this conjugation normally happens at the first carbon of the carboxyl group. You will see most of these modification reactions, they occur mainly at the carboxyl group carbon atom. Second is the decarboxylation. Again, since decarboxylation is there, it involves the COH group that is present on the jasmonic acid molecule. Glycosylation, again carboxyl group is involved in this kind of reaction. Then jasmonic acid further can undergo reduction reaction, and this reduction can either happen at the carbonyl group, which is present at the 6th carbon, or the double bond or the unsaturation that is present at the 9th or the 10th carbon. And it can undergo hydroxylation reaction, wherein a hydroxyl group is attached, and this addition of hydroxyl group can occur either at 11th carbon or the 12th carbon, and in some cases it has also been seen that the hydroxylation occurs at the 6th carbon in the jasmonic acid structure. So these are the various derivatives that are produced by the jasmonic acid, and till now more than 30 derivatives of jasmonic acid have been reported from the plants, and the major, or I will say the important derivatives are the amino acid conjugates, that is jasmonic acid isoleucine, or methyl jasmonate, wherein a methyl group is attached to jasmonic acid, when sugar is attached to the jasmonic acid, or the glycosidic form of jasmonic acid. Apart from that, certain minor derivatives are there, 11 hydroxy jasmonic acid or 6 hydroxy jasmonic acid, and so on and so forth. So these are the various derivatives or the modifications through which the jasmonic acid undergo and it produces different types of derivatives. Now we move on to the signaling pathway of jasmonic acid, that is how it produces its effect in the plant system. And before we discuss the mechanism, first I will discuss the important components of that signaling pathway. Now, for any molecule to act on a system, it requires certain receptors which bind that molecule, effective molecule, thereby producing downstream effects in the plant system. Same is in the case of jasmonic acid. We have jasmonate transporter protein 1 or JAT1, which are nothing but the receptors for jasmonic acid derivatives. And amongst all the derivatives, it has been found that amino acid conjugate, that is jasmonic acid isoleucine or jasmonate isoleucine derivative, is found to be the most effective amongst all the all the other derivatives that have been reported from the plants. So we can say that jasmonic or jasmonate isoleucine molecule binds to JAT1 protein, and from there the downstream signaling starts. Next component is the SCF complex, and SCF complex is nothing but it's a negative regulator of the jasmonic acid effects, or we can say it's a repressor protein. As the name indicate complex, it is a mixture of three different proteins. First is SKP1, that is S-phase kinase associated protein. Then Cullen protein, a ring box protein. Now ring box proteins are important proteins which act as a substrate for the ligation or ubiquitination of the 26S proteasome enzymes, which causes the degradation. And third component is the F box protein, which are specifically for the binding of the, or I'll say they are specifically for the ubiquitination of this complex, and F box protein example is COI1 or carnitine insensitive 1. So this is the second major complex or the component of the jasmonate signaling pathway. Next we have is the JAS1 protein or jasmonate ZIM protein. These are the ZIM is zinc finger inflorescence meristem, and again these type of proteins are responsible for controlling various growth and development factors in the plants. We also have JAI3 or jasmonate insensitive 3, which is also known as JAS3 protein. Again JAS1 and JAS3 protein are the repressor proteins which bind to the transcription factors, thereby inhibiting the expression of the genes that are stimulated by jasmonic acid to exhibit various physiological effects. So SCF complex, JAS1 and JAS3 are the repressor proteins which inhibit the or negatively regulate the jasmonic acid effect. And next component is the jasmonate insensitive 1 or JIN1, or it is also known as MYC2 protein. These are nothing but the transcription factors that stimulate or that initiate the expression, gene expression, which leads to various physiological effects induced by the jasmonic acid. Now what happens when jasmonic acid is not present? Now we come how these components work to produce the effect. I will take two conditions wherein in one condition no jasmonic acid is present, and in the second, that is the stress condition, where jasmonic signaling is produced or the jasmonic acid is present. So when jasmonic acid is not present, the genes remain inactivated. And how these genes remain inactivated? Because the transcription factors which activate these genes, they are inhibited by the JAS proteins. Now what do these JAS proteins do to inhibit transcription factor? They recruit GRCHO-TUP1 or type 1 co-repressor topless proteins, which are also known as TPL proteins, as well as topless related proteins, which are known as TPR proteins. Now these TPL and TPR proteins, they recruit another interactive protein which is known as novel interactor of JAS, that is ninja protein. And once this ninja protein is recruited, JAS proteins form a complex which is known as JAS ninja TPL complex, and this complex binds to transcription factors, thereby causing the inhibition of transcription factors. And once these transcription factors are inhibited, the gene expression is not stimulated or initiated, so no effects are produced by the no effects are shown by the plants, and those effects that are exhibited by jasmonic acid. Now the second condition is under stress condition, and it has been observed whenever the plant is exposed to stress condition, whether it is abiotic or biotic stress, this biosynthesis of jasmonic acid gets stimulated, and more jasmonic acid is being produced by the plant. And as I told you that jasmonic acid isoleucine derivative is the most active derivative. This derivative binds to JAT1 proteins, which are nothing but the JAT binding proteins or the receptors for the jasmonic acid or the jasmonic acid transporter proteins, and by binding these proteins they enter into the cytoplasm. Once they enter into the cytoplasm, they have more affinity for the SCF complex. So jasmonic acid isoleucine derivative, it immediately binds to the SCF complex, and mind you, SCF complex is nothing but a repressor protein which has inhibited the transcription factors. So once jasmonic acid molecules bind to SCF complex, it changes its structural configuration. Once its structural configuration changes, these changes increase the affinity for the Jas proteins, and immediately this complex recruit Jas protein for binding. And once Jas protein binds to this complex, it frees the transcription factors. So the transcription factors are now free. Now this Jas protein has Jas protein bind to SCF complex and the jasmonic acid complex, which causes ubiquitination of this complex and make it more susceptible or vulnerable towards the 26S proteasome degradation, and this complex undergo proteinaceous degradation. Now once the Jas protein undergo degradation, we know that Jas protein formed a complex which we call as Jas ninja TPL complex. So Jas proteins are now out of this complex and ninja TPL complex is free. Now this ninja TPL complex one gets free, it reduces its affinity for the transcription factors, and these transcription factors, they get free to exhibit or to induce the genetic expression. So GIN1 and or MYC2 transcriptional factors, they get free and increase the expression of the genes through another mediator intermediate protein which is known as mediator 25. Now this mediator 25 protein forms a bridge between the transcription factor and the genes which produces the effect of jasmonic acid. So this is the diagrammatic representation of this mechanism, and you can see that jasmonic isoleucine derivative, it enters into the plant system. Before it enters, ninja TPL complex binds to these transcription factor and causes the locking of the transcription factor, thereby no expression of the genes is there. But under stress condition, jasmonic acid through JAT proteins, that is jasmonic acid transporter proteins, enter into the cytoplasm, whereby it causes the ubiquitination of the Jas SCF complex protein, and this ubiquitination causes their degradation. Now once the Jas protein complex get degraded, ninja TPL complex is broken and they free the transcription factor, that is MYC2. And once this MYC2 is free to express, it stimulate or induce the gene expression, thereby producing various effects that are exhibited by the jasmonic acid. So this is how basically jasmonic acid produces its effect. Now we will discuss what are the various effects that are produced by the jasmonic acid. So as I told you that the biosynthesis of jasmonic acid increases during the stress conditions, and these stress can be both biotic or the abiotic stress conditions. So therefore we can say that jasmonic acid response first, and the prime response is to provide resistance toward these kind of stresses, whether it can be a wound response, it can be a attack of pathogens, it can be a salt tolerance effect, it can protect the plant against drought conditions, it can protect the plant against freezing conditions or any kind of light stress wherein different wavelengths of UV lights fall on the plant, thereby causing the induction of the stress, and immediately jasmonic acid synthesis increases and it provides the defense against that kind of stress. It can also protect the plant against the ozone, carbon dioxide or heavy metal toxicity stresses. So by and large you will see all the types of biotic or abiotic stresses when they occur on a particular plant, the jasmonic acid biosynthesis increases through its signaling pathway. It causes the expression of different kind of genes which produces the protective effect for that particular plant. Now apart from these stress conditions or protection of the plant against stress, the jasmonic acid has also been found to cause the elongation of the roots. It also leads to the development of the trichomes. Trichomes are again a sort of defense structure for the plants. Then it causes the repression of the flowering, because the first effect that of jasmonic acid that was observed was the flower senescence. So it causes the repression of the flowers. It also helps in the development of the stamens. It leads to the degradation of chlorophyll, since it leads to the senescence aspect. So chlorophyll degradation is there, no photosynthesis occur, leads to senescence. Then it also causes the accumulation of anthocyanins. So these are the various effects that are exhibited by jasmonic acid or its derivatives. Now I will just briefly discuss about some of the major uses of jasmonic acid or the effects that are produced by jasmonic acid. So first I will discuss jasmonic acid in the stress conditions, that is abiotic or the biotic. So as I have already told you that jasmonic acid production increases during stress conditions like high salinity is there, or drought conditions are there, heavy metal toxicity is there, or freezing conditions are there, or any kind of light or ozone stress is there, or any kind of pathogen if it attacks on the plant, the synthesis of jasmonic acid increases. Now this increased production of jasmonic acid further stimulate through its signaling pathway, further stimulate various antioxidant enzymes like catalase or superoxide dismutase or ascorbate peroxidases, and all these antioxidant enzymes, they basically decrease the lipid peroxidation. And once the lipid peroxidation is decreased, they prevent the disintegration of the cellular structures, thereby protecting the plant cells. Now apart from MYC2, jasmonic acid derivatives further stimulate various kind of other transcriptional factors like NAC, ERF or WRKY factors. They also activate calcium channel or the movement of the calcium ions inside or outside the plant cell, thereby maintaining the tonicity of the cell. They also activate MAP kinases, which sometimes also have a positive regulating effect on the jasmonic acid production. They increase the interaction with salicylic acid or abscisic acid or ethylene. So these interaction with other hormones also help in protecting or I'll say have a synergistic effect on the protecting effect of the jasmonic acid. Next I'll discuss about the effect of jasmonic acid in the stamen development, because it is another important effect of jasmonic acid. Till now we have discussed that jasmonic acid activates MYC2 transcription factor, and along with MYC2 it has been observed specifically in case of stamen development, it also activates another transcription factor, that is MYC5. Now this MYC5 along with MYC2, they also activate another type of transcription factor, that is MYB21 and 24. Now when all these transcription factors are activated, they form a complex MYC-MYB complex proteins, and these complex protein increases the expression of the genes that are responsible for the development of the stamens. So in the graphs you can clearly see the levels of the jasmonic acid or jasmonic acid isoleucine derivative, they increases during the development of the stamens, and once the stamens are developed or flowering stops, the concentration of the jasmonic acid or its derivative gradually decreases. We have already discussed that they have an impact on the expression of the genes which regulate different types of effects like anthocyanin accumulation. This occurs through a transcription factor MYB75. Likewise, it causes degradation of chlorophyll through a transcription factor FIL or YAB1 protein. Jasmonic acid also causes the root elongation through another transcription factor MYC2, 3, 4 and 5. So all these factors that are exhibited, they are exhibited through activation of various transcription factor. Likewise, it also provides resistance towards pathogenic bacteria and increase the leaf senescence through a WRKY proteins or 57 proteins. They also decrease flowering during the early vegetative development by stimulating another transcription factor, which is TOE1 or 2, and they lead to the development of the trichomes through the transcription factor MYB75, GL3 or EGL3. So friends, this was all about the jasmonic acid, how it was discovered, how it is biosynthesized in the plant, how through which pathway or what is the signaling mechanism of jasmonic acid, and how it exhibits its various effects in the plant system, and in the end we discuss what are the various effects of the jasmonic acid onto the plant system. Thank you.